The structure and organization of the eukaryotic chromosome is a bit more complicated than the prokaryotes. So, first of all, we now have a linear chromosome, and we typically have sets and more than one, and they are different. So remember, in the prokaryote, it was usually one circular, and if they had more than one chromosome, it was the same chromosome. Now we have linear chromosomes, and we know we have more than one chromosome, and they're usually different. Okay. The typical chromosome is tens of millions to hundreds of millions in base pair length. The genes are interdispersed, and so we have our gene region, we have several origin of replications. We know that there are many origin of replications, and they are about 100,000 base pairs apart. We also know that there's a centromere, and that at the ends we have telomeres. And the purpose of the centromere is to help us when it's time to pull apart our chromosomes. There are two different types of centromeres that exist. So there is a point centromere that exists in certain yeast species, and these are 125 base pairs in length. Then you have the ones that are found in humans, and these are regional centromeres. These tend to be longer. They're about 7,000 base pairs to over 1 million, and they're these repeated DNA sequences. But it takes more than just the sequence in the regional. So you also have this CENPA histone variant that replaces histone H3 that we'll talk about in a couple of slides. And so together, that helps to make this centromere functional. As mentioned in the keywords, the kinetochore is the proteins that bind to the centromere that allows for us to pull them apart during anaphase. And then we also know at the ends is these telomeres, and the purpose of these telomeres is to help stabilize us. So remember, if we didn't have these telomeres at the end blocking them, it would look like a double-stranded break, and the cell would want to work to put that back together again. It also helps with the end replication problem that we'll talk about during replication. And now throughout these chromosomes, you have repetitive sequences. It's also important to note that within the protein encoding genes, you have exons and introns. The exons are what's going to help to code for the proteins, and the introns have to be removed. So these are usually non-coding pieces. Okay. Now, if we go back to those repetitive sequences that I mentioned, okay? So, first we're going to describe what is known as this sequence complexity. And what does that mean? That is just talking about what do we have in what's making up our genome. So we know that we have a lot of repetitive sequences here. So if you look at this graph, okay, about 41% of our sequences are unique or non-repetitive, whereas the majority is repetitive. And that's broken down into moderately and highly repetitive sequence. So our unique or non-repetitive show up usually once, maybe a couple times. These tend to be our protein coding genes. There's our exons, our introns, and then sequences outside of the genes. Our repetitive sequences are found in our rRNA genes, as well as transposons, or those jumping genes we talked about, and at our centromere region. So we know that the majority of our genome is repetitive sequences. The majority of our genome is repetitive sequences that don't code for genes. Okay? And how much of a genome is repetitive sequence varies based on the organism. So, when we talk about all of this DNA, our DNA is over 1 meter long. A cell is 100 or 10 to 100 micrometers in diameter. A nucleus is only 2 to 4. So how do you fit something over 1 meter long into something that's 2 micrometers in diameter? The answer is compaction, like we've seen with the bacteria. So the first level of compaction is these nucleosomes. So you take the double helix of DNA and you wrap it around these histones. This gives the classic beads on a string that makes up chromatin, remember? And so now, when we talk about these nucleosomes, it's a histone octamer plus 146 to 147 base pairs of DNA. This histone octamer is composed of 2 copies of H2A, 2 copies of H2B, 2 copies of H3, and 2 copies of H4. Now if we back up a second, we just said at the centromere, this H3 is going to be replaced with a histone variant, CENPA, in the centromere region. Okay, so that's what we were referring to. So now, you take the double helix, you start to wrap it around the octamer. An octamer, which stands for 8, is 2 copies of H2A, 2 H2B, 2 H3, and 2 H4. Then you have a linker region, and this linker region is 20 to 100 base pairs. In this linker region, you find this linker histone H1, and there's also non-histone proteins that reside in this linker region. Are we okay with this idea? These proteins have a globular domain and an amino terminal tail in one histone. They tend to have positive amino acids that are exposed that interact with our DNA, because you should know that our DNA is a negatively charged molecule. Together, this shortens our DNA by about 7-fold. Now, after you have your nucleosomes, the next one is this 30 nanometer fiber, which is named because it's 30 nanometer size. This 30 nanometer size or fiber is this further compaction of our nucleosomes. Now, researchers knew that this could take on 2 ways. It could be this solenoid or a zigzag pattern. So, Timothy Richmond group in 2005 determined that it is in fact the zigzag pattern that prevails. Okay, so our 30 nanometer fiber has this zigzag formation. And now this also shortens the DNA by about 7-fold. So once we get here, before we get to our radial loops, our DNA is already shortened about 50-fold. So here in the radial loops, which look very similar to what we see in prokaryotes, we have this nuclear matrix, and this consists of the nuclear laminin and the internal nuclear matrix. And these are fibers that are inside of the nuclear membrane, and their job is to interact with what's in the laminin and what's in the membrane. And we know that we can anchor our 30 nanometer fiber to the nuclear membrane in these radial loops. And here you see our radial loops. If we zoom into what a radial loops look like, so this is going to be our 30 nanometer fiber, and we're going to make connections to the nuclear laminin. And we do this by using these sequences called MARS, or also called SARS. So what MARS is, it stands for these matrix attachment regions, also called scaffold attachment regions, so MARS or SARS, and they are interdispersed at different regions in the genome, and they bind to specific proteins in the nuclear matrix. So we have areas on the DNA whose job it is to bind to proteins in the nuclear matrix that allow us to form these radial loops, which allows us to further compact our DNA. Are we okay with this idea? Okay. And so then we know by staining that chromosomes occupy a certain area of the nucleus. So they're not just like the bowl of spaghetti like we think about, but it is actually each one has a certain part of the nucleus that is its own. So here we're looking at chromosomes in the metaphase and during interphase, and they have the same staining. And so this is typically what we're used to seeing. However, we know this is a small portion of what goes on in the cell, and the majority of the time it exists in the chromatin state. Well, in that chromatin state, they each occupy a different area. And this is important because now proteins essentially know where they need to go to transcribe certain genes. So we have the radial loops that sit inside of the nucleus in defined areas that belongs to each chromosome. And this is a chromosome territory, okay? So each chromosome in the nucleus has its own distinct area that it occupies. And it's also important to note here that the compaction of the interphase chromosomes is different. So this goes back to our discussion about euchromatin and heterochromatin. Euchromatin and heterochromatin during interphase has different compaction. The euchromatin tends to be looser here, whereas the heterochromatin is more compact. The euchromatin is looser so that proteins can get to our DNA and it can be transcribed. So when we talked about transcriptionally active versus inactive, so the heterochromatin tends to be further compacted and not as active because it will take more for our proteins to get to those areas. Okay. Now everything we just described is how we compact an interphase chromosome. Now we're going to talk about what happens in a metaphase chromosome. It starts out the same. So you have this double helix DNA molecule. You're going to wrap it around these histone octamers to create a nucleosome, which is the 126 or 7 base pairs of DNA wrapped around 8 histone proteins called an octamer that has 2 H2A, 2 H2B, 2 H3, and 2 H4. Then we take that and we form our 30 nanometer fiber, which has the zigzag pattern. We take our 30 nanometer fibers and we attach it to the nuclear laminin at the Mars site. Remember that? Now what happens next is we take these radial loops and we further compact them together. So we take the radial loops and we loop them out. So in this sense, just looping the loops is how we're doing this. And when we loop the loops, it gives the characteristic metaphase chromosome that we're used to seeing. So instead of taking the radial loops and giving them their own chromosome territories in interphase, we take the radial loops and further compact them, loop them further to get the classic metaphase chromosome, or the X that we're used to seeing. This is a highly compacted piece of DNA. This is usually not that transcriptionally active. So remember that metaphase would be an hour out of the 24-hour day if that was the cell cycle in this. And it only stays here for a short period of time. And when it's here, it doesn't transcribe many genes. There are some genes that are able to be transcribed here, but not many, because it's so compacted, proteins can't access it. Okay? So we just talked about the DNA in eukaryotes. Now you can go back to your chart and you can fill this in, and you should have enough information to now complete your chart. If not, if anything is blank, come to class and we can talk about it. Questions, comments, or concerns?